Genedrives are here.
Researchers have micro-injected mosquito eggs with DNA carrying the lethality gene, overproducing a protein that clogs up the machinery in a female mosquito's cells, preventing essential genes from being expressed and eventually killing them.
A year and a half ago, I made a video series called The Final Revolution. That video had a heavy emphasis on the eugenics device Genedrives, and it stated that they were more dangerous than nuclear bombs. This is because a gene drive doesn't just spread through a population at a normal rate like a normal gene. A gene drive is a form of genetic engineering that, instead of a 50% inheritance rate like a normal gene has, it has a 100% inheritance rate, meaning once it's established in a species, it cannot be removed from that species until everybody carrying it is made extinct. And as long as one survivor remains, that gene drive can spread through the entire population, and in some populations, that means very, very quickly. This can have all sorts of dramatic effects, up to and including the extinction of entire species.
And if they were to do that and let that fly escape outside, there's a non-trivial chance that it's going to find a mate, and most of the offspring are inherit. And if enough of them survive to mate, and their offspring inherit, then the odds that it will, by chance, go extinct become low enough that it will eventually spread through the local population around wherever this was released, and then further, assisted by human transport, all around the world, potentially to every population of that species, whatever that species is.
CRISPR, genetic engineering, allows gene drives to be implanted anywhere and to do just about anything. CRISPR gene drives were created at MIT by Kevin Asvelt at the MIT Media Lab, the subsection, the Program for Evolutionary Dynamics. Now, the Final Revolution series also focused on Jeffrey Epstein, who claims to have founded the Program for Evolutionary Dynamics. And there was a big cover-up where Bill Gates was funding this program in secret, and it was hidden with the president of MIT's help, and it was a whole big crazy thing. The official founder of the lab is Nicholas Negroponte. Nicholas Negroponte, his brother John, was the ambassador to Honduras accused of facilitating a coup, and the first director of national intelligence in the United States under George W. Bush. Director of National Intelligence is the head of all U.S. intelligence. By the way, Wired magazine is also founded by Nicholas Negroponte, the guy who claimed to have founded the same lab that Jeffrey Epstein worked at and claimed to have founded, as well as funded. So when the Rolling Stones did an article recently looking into was Jeffrey Epstein a spy, well, it's funny that they didn't mention that he worked with the brother of the Director of National Security. It's either some huge incompetence or willful ignorance. British scientists have already implanted gene drives in human embryos on the promise that they would destroy the embryos. Researchers from the University of California have developed a gene drive to exterminate the Asiatic fruit fly. Gene drives have been proposed to exterminate ticks that carry Lyme disease. Kevin Asvelt's method to eliminate Lyme disease in ticks is actually to vaccinate them, or to synthesize a genetic vaccine and then inject mice with it and use CRISPR gene drives on mice to eliminate Lyme disease in ticks. And this would kill any ticks that bite the mice. And they're talking about releasing this Nantucket and Martha's Vineyard. Now, if a tick can be made to die from biting a mouse, what happens when that tick bites you? You see where I'm going with this concept. What happens when that mosquito bites you? And they've been proposed for use in the New Zealand Predator-Free 2050 program. That's a program to eliminate 8 different mammalian species, including rats, types of weasels, some possums. It's pretty crazy. The New Zealand government, though, is pretty crazy too. As the creator of CRISPR Genedrive says, in our view, it is wise to assume that invasive and self-propagating gene drive systems are likely to spread to every population of the target species throughout the world. So we could wake up in a couple of years and go, hey, where'd all the rats go? And looking even bigger, here we find Bill Gates again. Target Malaria, a project funded by the Bill and Melinda Gates Foundation, invested $75 million into Genedrive technology. The foundation originally estimated that the technology would be ready for field use by 2029 somewhere in Africa. However, in 2016, Gates changed this estimate to sometime within the following 2 years. 2019, auspicious year or what? In December 2017, documents released under the Freedom of Information Act showed that DARPA, Defense Advanced Research Project Agency, had invested $100 million into Genedrive research. Is that the same project? It's not really clear. Either way, not good. DARPA should be investing in preventing gene drives from spreading uncontrolled, and actually, one of my early theories was that that's what the lockdowns were about. And maybe this new virus going around was a viral vector therapy with the gene drive strapped onto it.
As spring arrived in the Florida Keys this year, mosquitoes began emerging from boxes just like these. These particular Aedes aegypti mosquitoes are all male and engineered to carry a gene that kills female offspring. That's because it's the females that bite and thus contribute to the spread of diseases like dengue fever and Zika.
So when I say gene drives are here, I don't know of any that have been actively deployed yet. Some people have said they have been. I think there's some confusion in that the Oxitec mosquitoes are at least supposedly genetically modified mosquitoes, but are not gene drive mosquitoes. So they can be bred out over time, as this video says, 10 generations.
Keys mosquitoes all have a fluorescent marker that allows Oxitec to monitor them and their offspring for up to 10 generations. And they say only the Aedes aegypti mosquito is affected, and that the impact on the Keys ecosystem will be minimal.
So there are no harmful impacts for beneficial insects like bees or butterflies, and even other mosquitoes.
From the Global News article, by genetically altering the DNA of mosquitoes, scientists hope to prevent them from passing on pathogens to humans and therefore control the spread of vector-borne diseases. 2 years ago in the town of Bana, a 4-hour drive from Burkina Faso, researchers released thousands of genetically modified mosquitoes. Then, allegedly, they collected all of those mosquitoes, which I call bullshit on.
But risk is a difficult thing to calculate, which is why Carrie Bowman stresses the need for informed consent, even if cultural differences might make it harder to obtain.
Yes, then it must be said that here we have abused local communities by simply explaining to them that it is research that will overcome malaria. But we did not explain the health, economy, political, and ecological issues behind this. We have not made it clear to the community that we are going to manipulate mosquitoes genetically. Besides, it is difficult in our language to describe genetic modifications.
Apparently that's not the only such test that's been happening. The West African country has been a testing ground in recent years for biotechnology such as genetically engineered mosquitoes. I guess they're just dumping all sorts of shit over there. Yet laws regulating genetically engineered species aren't consistent between countries, despite their ability to cross borders, right? With crossover to the entire planet. They don't even know how to classify them. The U.S. administration, they classified them as a form of pesticide or a fungicide, insecticide, or rodenticide. To me, it seems more like a national security risk, a global security risk. It's not an insecticide, it's an animal, says Barry Ray, the executive director of the Florida Keys Environmental Coalition.
Researchers have micro-injected mosquito eggs with DNA carrying the lethality gene overproducing a protein that clogs up the machinery in a female mosquito's cells, preventing essential genes from being expressed and eventually killing them.
So this company, these genetically modified mosquitoes by Oxitec, they add apparently a lethal gene. Gotta love the scientific and technical terms.
Oxitec, the company behind the experiment in the Florida Keys, has released mosquitoes in Brazil, Panama, and the Cayman Islands. Here, scientists use the gene editing tool CRISPR to insert an enzyme that cuts the X chromosomes during sperm production.
We barely understand ecosystems, and we could study ecosystems for another 100 years and we still wouldn't fully understand them. So this is the real risk here, is we don't fully understand ecosystems.
And they just said that it was super remote, that there would be no females released, and so they didn't even test what would happen if, these female mosquitoes bit people. They just didn't properly test it.
Oxitec has received approvals from the Environmental Protection Agency as well as state and local authorities. But the head of the Florida Keys Environmental Coalition, Barry Ray, points to the EPA's risk assessment, wherein the agency noted it hasn't studied the health risk from a genetically modified mosquito bite because the likelihood of a female surviving is negligible.
Okay, well, that's great until you find this out.
But Oxitec uses the antibiotic tetracycline as a switch which can halt production of the protein that kills the female offspring, meaning the females will survive. It's used in their laboratory in the UK to keep females alive long enough to lay eggs. And Barry Ray says it can be found in sewage in the Florida Keys.
We have a lot of abandoned septic systems down here. We have active septic systems still. We have open ports to a vacuum-based sewage system. So there's cans of water sitting around where mosquitoes can lay their eggs at the edges and perhaps hatch mosquitoes. But to not investigate that brings up one risk, that you could be producing female mosquitoes and not even knowing it.
Yeah, their switch is tricycline, a common antibiotic that is in sewage water. I mean, hell, maybe somebody dumps their pills into a puddle. And next thing you know, especially if this is public knowledge, somebody could consciously do this. And for your gene switch to be a commonly used pharmaceutical, it's absolutely insane. Totally insane.
Oxitec and the Florida Keys Mosquito Control District both say if genetically modified females are found, they'll halt the experiment.
And we have committed to the EPA as part of the permit that if there's any females released, that the process will stop right there until we get it sorted out.
And here's where it gets confusing. So that is Oxitec, and I've seen my videos went over a lot of national security lectures where they talked about this stuff, and I mean, they don't know what they're doing. They don't know how to handle it.
Authority can't keep up. It's just too slow. So we've been trying to do these stopgap measures. We've been first trying to let all the scientists know about the danger. I spend a lot of time talking to iGEM, which is an international genetically engineered machines competition, a bunch of talented undergrads and even high school students who play about with genetically engineered organisms. Very important that they understand that they should not insert the CRISPR system and instructions for targeting it into the same place that it targets. Because you do that in a sexually reproducing organism, you just made a CRISPR gene drive, boom, gone. Now, yes, you run the risk that one of them is going to do this deliberately. Eventually that's going to happen, but we have to push it off for long enough so that we can do some good with the technology first.
At what point is this informed consent, right? How much do they really understand what's going on? And how much are they going to turn around and blame you if something goes bad by saying you didn't explain it well, is one piece. And then also, where's the government in all of this? The regulators are probably not able to keep up with all of this. And the reason we love you and we wanted you to join the Media Lab was that of all the scientists who work on this stuff, I think you're the only one at this level who is thinking about the deployment risks and structures and informed consent and everything that we talked about, and the safety stuff.
The strong version of the precautionary principle says don't do anything until you can understand all of the consequences. And that's fine as long as you get to stay where you are if you do nothing. But that's not the world we live in. It might seem that way to some people, but that's not the world we live in. Civilization is not sustainable. We need continued invention. We need new technologies because otherwise we're eroding our natural resource base and we have to invent our way out of it. There's no other choice. We've passed the point of no return. There's too many people. We need to be more efficient. We need new technologies, and they have to be ever more powerful. But that's a double-edged sword. How do you ensure that we use them wisely? How do you ensure that we don't accidentally open a technological box that destroys us all? They're out there. There are some in biology. I'm not going to talk about it further, but they're out there.
Sure, happy to. I mean, that really gets to the crux of the FBI's outreach efforts, is that we stand on the premise that the scientific community is already incredibly ethical and responsible.
And this is, in principle, easy enough to do that a lot of people have the ability to engineer, say, a fruit fly to do this. And as long as people want to use this, eventually someone will. That is to say, if it is possible for a small group of scientists to build something that could take a decent crack out of malaria, someone's going to want to be a hero and release it without consulting everyone else.
Ray's other concern is off-target mutations that could result in a mosquito that is better adapted to transmitting pathogens, more aggressive, and that mosquitoes raised near antibiotics could become more chemically resistant. It could be years or even decades before we fully understand the implications of these projects.
At present, there are no guidelines that speak specifically to Genedrives, and we'll discuss that in a few minutes. But relevant U.S. guidelines and technical documents are not yet sufficient on their own to guide ecological risk assessment for Genedrive technology. This is a gap that we did identify. But we concluded that existing mechanisms of governance may be inadequate to address potential immediate and long-term environmental and public health consequences because they don't consider Genedrive's intentional spread. They don't consider Genedrive's potential irreversible effect on ecosystems. Often they lack clarity in the jurisdiction of their oversight and who it is that is responsible for which components of oversight. Most of them provide no structure for public engagement, and those that do have insufficient structures for public engagement. And they don't address the potential for misuse.
So the Oxitec are genetically modified. Target malaria, though, those are the Genedrive mosquitoes that Bill Gates is funding, that DARPA is funding. So are these genetically modified, or are these Genedrive mosquitoes? In Burkina Faso, researchers with target malaria have evaluated the data from the initial release of sterile male mosquitoes. Okay, so this is important. In male mosquitoes, Genedrives will cause a population collapse. It's when they enter females that it causes a complete extinction of the species. So they released sterile male mosquitoes. Maybe that's their trick there. Sterile male mosquitoes. They released them, apparently they were sterile, and then they collected them all again. So, maybe that's the caveat here. They're not releasing Genedrive mosquitoes that are able to breed. But man, we're getting really close. So the laboratory partners are now working on a mosquito that can reproduce with only male survivors. The goal is to eventually release Genedrive mosquitoes, which would allow a gene to pass through generations of pests and ultimately lead to a population that is predominantly male or where the females are sterile. So, complete extinction of the species, at least, of the mosquito, if not mosquitoes as a whole, which is a perhaps small possibility, perhaps larger. Who knows? We haven't done proper testing yet.
It's one of the reasons why we are, the FBI has been engaging the amateur biology do-it-yourself bio community, sponsoring undergraduate synthetic biology competitions, which are now leveraging CRISPR-Cas9 in their projects. The window of opportunity for engagement, it exists now, and we need to capitalize on it before that particular horse has left the barn. I mean, I think we're already at that point.
This is Liz Heitman. This is Wayne Landis. I have a question.
Go, Wayne.
Okay, Wayne.
So, the detection of the use of these materials, or the prevention of it, seems like it's going to be problematic because so many of the research laboratories can be dual used. Whether it's an agricultural research area in some faraway place, something in the United States, it just seems to me that the detection of the potential use of these materials in warfare, I think that's going to be a huge challenge. And you guys didn't address it too much other than saying we should be ethical. But from my experience working at the Edgewood Area Admiralty Proving Ground, I know this can be enormously problematic. My example is if you can make an organophosphate pesticide, you can make a chemical warfare agent. And to distinguish between those two things is very difficult. And I'd like to hear some of your comments on that.
Ed, would you like to get it started?
Sure, happy to. I mean, that really gets to the crux of the FBI's outreach efforts, is that we stand on the premise that the scientific community is already incredibly ethical and responsible. The challenges that and the need for the education component is providing security awareness. That the whole dual use argument is not going to be list-based, but it absolutely is going to be looking at intent. And it's very difficult and challenging for the community as it stands now that are not already under some sort of regulatory regime to be aware of what some of the security implications are. And that's why I stated early on that now more than ever at the undergraduate or even high school level, where some of these tools are becoming more accessible.
The undergraduate or even high school level, incredibly ethical and responsible.
Liz or Jim, might I ask one more question?
Yeah, sure, Wayne, go ahead.
So I actually sat here and I just went through the entomological terrorism, a tactic, and asymmetric warfare. And my specialty is risk assessment, and it talks about risk management and assessment. This paper doesn't have any kind of quantitative aspect to it. And I'm trying to get a feel for what's been done as it comes to looking at a report and so forth. I was wondering, has there been actual quantitative studies done on what the particular outcomes are and if that's available? Because, I'm looking through this and there's lots of words but no numbers. To me, it's probably not very real until there are numbers with it. So I guess I'm asking you guys, are you aware of efforts to put numbers to these assertions? And if those things are available?
This is Dr. Dalge. I have not seen any numbers put onto it. That paper is the only one that really comes up when you look. I don't know. Yeah. There may be colleagues in the Department of Homeland Security that do the risk assessment that might have gamed this out or thought about this, but I haven't seen anything myself.
Great. Thank you very much.
Right, and this goes back to my the project that the FBI did jointly with the National Academies, and the fact that there is right now no effort to do not only what you're asking, to put numbers to that specific scenario, but just taking a step back and doing an overarching assessment on what the impacts are on something like Genedrives or other similar types of applications.
Says, an activist from Burkina Faso, we've never done a national survey for this project. You will see organized marches where we have several hundred organizations that are against this project. This project is controversial, but that's never stopped Bill Gates from experimenting on Africans before. In Canada, genetically engineered mosquitoes, whether they include Genedrive technology or not, would be regulated under the Pest Control Products Act. But Canada says there are no plans to release such technology currently. I guess to solve all the endemic diseases that mosquitoes spread in Canada, right? With the release of genetically engineered mosquitoes still in the experimental phase, we're still a long way off from determining whether they would even be effective as a weapon against disease. Which is interesting because let's look at this clip here.
In the Keys, diseases like dengue and Zika are a real concern for authorities. Last year, 70 cases of dengue fever were reported in Florida. So how could the gene editing of mosquitoes help suppress vector-borne diseases like Zika or West Nile? And what are the potential drawbacks?
Okay, so we've got, for one, we've got 750 million genetically modified mosquitoes dropped off in Florida. And why? Well, to prevent dengue fever in Florida, right? Well, you've got cases every year of dengue fever that break out in Florida. So I decided to check that out as well. And hey, look at this. 0 of those cases in the last year actually were contracted in Florida. Every single case was contracted outside of Florida, making those genetically modified mosquitoes a little questionable. Especially how are you going to prevent a disease that's not coming from your area? There are suspected cases of the fever being contracted inside of the state in previous years, but it's very small scale, and to risk so much for so little, I don't know, seems crazy to me. Especially what New Zealand's doing, getting rid of the rats. I guess Jacinda thinks they're icky. Genedrives are something that everyone in the world should know about. They're more dangerous than nuclear bombs. I bet you if you asked 95 out of 100 people in North America what a Genedrive was, or in Europe what a Genedrive was, they would have no idea. Burkina Faso, what do you think the percentage is there? And yet, they're coming. They're apparently already here. And it can get a lot worse than mosquitoes. And one day we're going to have to deal with it.
So the first half will be towards the beginning where I talk about reading DNA, because reading and writing DNA are very heavily interconnected. And the second point where we're talking about humans 2.0, I'll mention it. This is part of our efforts to reverse aging in humans. So I've been involved for over a decade with companies in China.
People look to George Church. He's got gravitational pull in the genomics field.
There's kind of this public world, and then there's what's actually happening.
Is something I'm going to do or is something I'm going to sell going to hurt somebody? I've never thought about it.
Anybody can use gene editing.
I've been here for a week and I found 3 people editing human embryos.
Our dream team from left to right. First, George Church is professor of genetics at Harvard Medical School and director of personalgenomes.org. His 1984 Harvard PhD, and why is it always 1984? Lee Silver, he's the author of many publications, including Challenging Nature and Remaking Eden, Cloning and Beyond in a Brave New World.
I suspect that people will separate sex from reproduction.
And yet, if Lee is right, that this is going to be the way that we reproduce in the future, or a certain set amongst us.
We now can take blood from the arm of a pregnant woman and essentially sequence the whole genome of the fetus.
But insurance companies and governments will have a massive incentive to screen all embryos, again, because it'll cost next to nothing. But let's say then you have 10 fertilized eggs. You'll be able to do a full genomic analysis on each of those 10 through PGD. Through stem cells or induced stem cells, we'll be able to generate larger numbers of eggs, because right now human eggs are a limiting factor. And then if you have 1,000 eggs that are blastocysts that are fertilized eggs, and you're doing this probabilistic analysis of each of them, it really opens up a lot of issues and a lot of possibilities.
So there are genetically modified humans. CRISPR is just one of many technologies that's particularly promising because you can rapidly prototype and precisely change, not just add in an extra copy, which is what typically gene therapy has been, but you can alter specific places. Now, back to Bob's point that we can screen for embryos, well, that assumes that we have, if you start screening for a large number of things, if you screen for one thing, then about half your embryos are okay. If you screen for two things, it's going to be 1/4 and so forth. You get up to 10 things and you don't have enough embryos. So the alternative would be to genetically engineer them to be whatever you want them to be.
Is something I'm going to do or is something I'm going to sell going to hurt somebody? I've never thought about it.
Whenever you talk about biotechnology and the revolution, there's this famous slide that the National Institutes of Health generated, where it shows Moore's law, which shows that the power of computer transistors and chips would double every 2 years, while the price would be cut in half. And so that makes this extraordinary looking trend line on the graph. And then below that is the cost of genomic sequencing, which is tracking along at $100,000. Well, it starts at $3 billion, then it comes down to $100 million, and then it gets down to about 25,000, and then it just falls off the cliff about 2007, when next generation genetic sequencing was invented, which George, of course, was deeply involved with. And so that implies that the innovation response to those dynamics could be multitudes greater than the innovation that's followed because of Moore's law.
The plug-and-play analogy, the analogy that DNA is just a code, very popular in Silicon Valley. I think a lot of people would like to use biology the same way they used computers, to make piles of money, to change the world. I don't know how far the analogy holds up.
Josiah, you have been known for doing some pretty radical things with this technology. So the first time I heard about you, you had done your own fecal transplant. Then you were doing, making your skin cells glow, or trying to make your skin cells glow. I'm not sure you pulled that off. And then I actually saw you inject a plasmid with a myostatin knockout, which, if it worked, would have allowed your musculature to continue growing. So I guess the big question I have for you is, what is your role in this neo-biological revolution?
Yeah.
Why do you do what you do?
I kind of see myself as an activist and an advocate. If you ask somebody right now, could you buy a kit online, have it shipped to France or China or someplace in the US, where you could do genetic engineering, where you could use CRISPR, most people would have no idea. They would be like, that exists? I can do that in my home? It's not illegal? In some countries in the EU, it is illegal. But it does exist, and people don't know about it. How do we make this available to everybody, right? I'm a big advocate of self-experimentation, of gaining knowledge and doing things to push science forward and trying to get around the system, because the question isn't anymore, how are they going to regulate us? It's how are they going to stop us? We've been doing gene therapy on humans since the late 90s. We know it works. We know you can modify humans. This isn't something that people should question anymore. It works. That's what I want to tell people. That's what I want to let people know. That's what I want people to do. Modify yourself.
Okay. 4 people agree with you.
There's literally a cure for a disease using gene therapy that's out there that's not available. And you know what? I read through the regulatory filings, the patents, and the papers. You can reverse engineer that. I can do gene therapy piracy, provide that to you for a couple $1,000s to cure your disease. Why is nobody doing this?
Right now we are in the 2nd International Summit of Human Genome Editing in Hong Kong. basically we are waiting for Dr. J.K. Hayes' talk. They obtained about 30 eggs from these 7 couples, and they actually impregnated one couple with 2 embryos, edited embryos, and now we have a twin girl, Nana and Lulu, that are born.
The general feeling amongst the scientific community is that we shouldn't go ahead yet.
An immediate outpouring of frustration by the scientific community. But George, you were one of the lone voices to defend him. I think a lot of subsequent information has come out about, or questions have come out about it. You know, was he addressing an unmet medical need? Did the people participating in that actually have truly informed and meaningful consent? And is the data valid? And did he execute well? So, in fact, Feng Zhang famously said that he didn't actually share, even though he consulted with scientists, he didn't actually share his intentions with Feng Zhang after he criticized him for his technique. So I'm just curious, do you still defend J.K. Hayes' actions?
I didn't defend him so much as say that there should be a balanced discussion and there should be a focusing on the data. So many of the reactions occurred before anybody had seen it. In fact, they still haven't seen the data. I had access to the data prior to the meeting.
Did you know what he had done?
I did. So there were 2 papers that were provided to me by the Associated Press. So I felt that I met my responsibility of communicating with the public the threat. While there were other people who knew that he was doing it, they didn't have access to data, but they knew he was doing it, they did not report, did not feel a desire or a need to report what he was doing. And I think that was the failure of the system. It was not that we didn't have we had a moratorium, we even had laws and consequences, but everybody was ignoring them. But
CCR5, knocking it out can help prevent HIV.
Yeah.
Excuse me. But it can also enhance our cognition.
I mean, I don't think that
Is this a front for a brain enhancement experiment?
It is a form of enhancement. I mean, I think one of the critiques of why would you do the experiment is that the medical need for gene editing of human embryos and making people is not that great, or it's not clear where it is. There might be some sort of very narrow cases where it's medically required. Yet it's interesting technology that could work, so it kind of very quickly opens this horizon of enhancement. I think trying to make someone immune or resistant to HIV is an enhancement. Just like getting a vaccine, someone mentioned smallpox vaccine, that's also an enhancement. So very quickly, and it's interesting, you know, the first case is already an enhancement case. So I think that's where the conversation goes. You know, what kind of enhancements could we get, and do we want? There could be enhancements. George has a kind of a whole list of genes.
That's right. Let's talk about that.
You know, there's one that I wouldn't mind having, which is protection against cognitive decline. I don't know what cost that particular gene mutation has, but there are people out there who they found them in Iceland. You know, if you have a specific mutation in a specific gene, people live, you know, with clear minds until the end of their lives. And I think, you know, in our society where people live longer and longer, you know, that's a kind of enhancement that one would seriously consider, just given even the cost of caring for old people and that kind of thing.
And those enhancements could be done in adults, not just in embryos. So I think when we entangle those, we're unnecessarily giving reassurance that if we block the embryos and we block enhancement, that's just not true.
So Josiah, you talk mostly about enhancing ourselves, right? Is that where you see this going? Are you more comfortable with that kind of an approach? Or what is your take on human embryo editing?
We shouldn't be worrying about what is a medical condition and what is an enhancement. What we should be worrying about is how do we get everybody access to this technology, right? The only reason we're worried about enhancements is because we're afraid that the people who can afford them are going to get all these enhancements and the people who can't afford them aren't. And it's going to create a huge class distortion. Well, if we come into it, like George said, and we try to make this stuff affordable, if we start from the bottom and say, let's focus on access. Why is nobody talking about access? All people are talking about is, how do we outlaw this? How do we do this and do that? There's 40 countries in the world with no medical regulatory body. You can't stop this. Nobody can stop this.
I mean, last week we had a call for a moratorium on creation of more genetically modified people from a bunch of senior scientists around the world. And so I think the discussion, I mean, just in contrast to what you're saying, Josiah, the contrast is actually about more government control. That's where that conversation is today, this week. It could change. So there's these different currents. And a lot of people, not George, but other people in the scientific community, I think they're kind of calling in the regulators and the governments to, put the screws on this.
How do you stop it, though, right? Like, I could order this stuff to my house. I could order a microinjector on eBay. Companies do single-cell sequencing for me, right? The only thing I need is somebody to retrieve embryos, get sperm, and do the implantation.
Yeah, I have to cut a check, I bet, right? It's who you know. That's how you live forever. It's who you know.
Yeah.